My name is Krista Milbury, and on behalf of the Foundation for Conservation of Atlantic Salmon, I would like to welcome you to today's webinar. If you wish to use translation, please navigate to the chat where you will find instructions on how to connect for the AI translation. For today's webinar, we are very pleased to be hosting Dr. Mark Fast. Dr. Fast is a professor of fish health and comparative immunology at the Atlantic Veterinary College of UPEI. His research group focuses on how the environment impacts fish immunophysiology and interactions with their pathogens and parasites. For over 25 years, Dr. Fast has worked on the impacts of sea lice on wild and farm salmonids of North America, and this work has led to his group's involvement in multidisciplinary initiatives in Chile, UK, and Norway, next-generation sequencing techniques, Crispr and comparative genomics to examine novel intervention strategies to monitor drug resistance and combat infections. While the majority of the team's work involves tackling health issues of cultured fish species, Dr. Fast also has ongoing research projects in species of conservation concern. Today, Dr. Fast will be speaking on harnessing comparative genomics and CRISPR for sea lice resistance in Atlantic salmon. After the presentation today, we will be opening the floor for questions and answers, and I will now turn the webinar over to Mark.
Thank you very much. It's a pleasure to be here. You can see it's a very long title and very many contributors to our research. For those of you who are not super familiar with sea lice, they're not good for a whole lot in terms of if you're a salmon or you're interested in salmon biology. There are marine ectoparasitic copepod that impact obviously the marine phase of the salmon life cycle. Just giving a couple examples of two particular species of sea lice, Lepidopterus salmonis and Caligus rogercressii. Their life cycles are on the right of the slide here. These two parasites impact aquaculture in Lepidopterus salmonis in the North Pacific and North Atlantic, and Caligus rogercressii in the South Pacific in Chile. So they're controlled, and many different intervention strategies are used in aquaculture for that 18 to 24 months while salmon are out at sea. The biggest, or the kind of the way it essentially occurs, is that spillover infections from wild fish in the spring tend to increase on the farm through fall, and there's little recruitment in the winter at less than 4 degrees. So very few lice at those temperatures, and then they really increase in the second year of production, and there's a major concern for spillback of lice back to the wild populations. And of course, Atlantic salmon, unfortunately, are very susceptible. We have a picture here in the bottom right of an infected pink salmon. The concern on the Pacific coast is the very small size of salmonids as they go out to sea and pass aquaculture farms and potentially get exposed at unnaturally early life stages. So while the Atlantic salmon, of course, is interest for FCAS and for us, we all know that wild Atlantic salmon are in trouble. I won't go into it too much today, but this is actually some data from an FCAS sponsored research that we've been doing in Newfoundland, looking at impacts and infection pressure of sea lice farms on wild Atlantic salmon in two particular river systems, the Conn River and Campbellton River. And if you look at the graph on the left, and you could probably say this for a lot of Atlantic salmon populations, you see a strong decrease in number in returning salmon from the mid-late 1980s through to present day. And our study there, you can see on the right side of the slide, are looking at both releases from the river system and sentinel cages to assess lice infection pressure there. But with or without sea lice, wild Atlantic salmon populations are definitely not where they used to be. In terms of aquaculture, I mentioned that sea lice are controlled or they're attempted to be controlled globally wherever salmon are farmed. And if you look from the 80s and 90s, many of the, well, throughout agriculture's history, treatments that are used to control sea lice have been repurposed from agriculture, essentially. So in the 80s and 90s, organophosphates and pyrethroids were used. And then in the late 90s, early 2000s, and ivermectin, emamectin benzoate was sold under the trade name Lice. It's sold under that trade name, and it was quite effective for those years. But you can see from the graphs here in Norway, not so much in BC, but in Norway, and this would mirror what we, or this would be exactly what we see or parallel what we see in Atlantic Canada and the UK, is that around 2006, 2007, or 2008, the amount of drug use to control infections and salmon farms really started to increase. And that actually occurred around the time that the lice treatments were beginning to be ineffective. So you can see the line, there's a black line for salmon produced, and that's not really changing too much, but the red line shows that each year from 2008 up to 2014, there's an ever increasing amount of salmon that are being treated. So when the lines are equal to each other, that would essentially mean there's one treatment per production cycle. By 2014, you're getting 7-8 treatments for production cycle in Norway. And we were seeing the same thing in Atlantic Canada. BC, however, has a very different relationship in terms of salmon farming and wild salmon. And the lice infections have never been as bad in British Columbia, and so treatment was never needed as much in BC compared to the rest of the world. What happens with these treatment trends when products begin to fail? So in 2008, 2009, there's an example on the right of Atlantic salmon. You can see in a sea cage, they have these red open lesions and sores on their head, and that's all from sea lice as they aggregate towards the head region. You can see the precipitous decline in drug use in Norway for salmon farming after 2014, and it stayed the same. The data from 2016 and 2017 is about equal to what it is now, so it's kind of plateaued. But those treatment data really dropped off, and that wasn't because there was more effective control or that there weren't as many lice. It was just that, the drugs that were being used weren't working. There was a major shift to doing non-medicinal and mechanical treatments for sea lice. We've actually been one of the groups trying to monitor and test for emamectin benzoate resistance globally. There's a picture of our student, Shelby Reimer, who's been doing whole genome sequencing out in BC to assess emamectin benzoate resistance in sea lice, and been doing some work with Ian Bradbury's group in the Atlantic. And essentially, this has shown that in areas both in the Pacific and the Atlantic where resistance to this drug has occurred, we've actually found changes in the louse genome. So the complex sort of graphs you can see on the right are Manhattan plots of the 16 chromosomes, 15 chromosomes plus one sex chromosome in Lepidopterus salmonis. And the highlighted area in the middle is the changing region in chromosome 5 associated with emamectin benzoate resistance. So we're trying to develop a test for industry so that they understand populations that are resistant to emamectin and don't use that treatment. And essentially, this is all data has come from work being done on bioassays. You can see a bioassay down the left where lice are treated in a dish, and we identify concentrations of the drug that they're susceptible to, and that's been increasing over time. I mentioned that in BC, things are a little bit different than we see in the North Atlantic. So there are many reasons for this, but in terms of resistance to or lack of developing resistance to certain drugs, one of our former postdocs, Gregor, actually was able to show that large wild populations can actually act as a refugia from the treatment. And so the larger the wild population, while that may cause initial infections on the farm, because that's where the lice come from initially. Because fish in the wild aren't treated with any medicinal treatments, the lice that they're infected with don't develop resistance to them. And so the lice on the farm actually also don't develop it, because there's always an exchange of wild and farmed lice. And so the graphs on the right show a proportion of resistant alleles over time. And in the top and bottom graphs, the populations with no or very few wild salmon, you can see the proportion of resistant alleles increase very quickly. And that's exactly what we observed in the North Atlantic for emamectin benzoate. Whereas if you have decently sized wild population, the resistance doesn't fix in the population, it doesn't develop. Despite all that, there's been some data over the last couple of years suggesting that these bioassays are showing increased tolerance in lice populations, both male and female, in BC, and that there's an increase in post-treatment lice numbers. So you can see on the far right of both of those graphs, they're starting to trend in the wrong direction and up. So emamectin benzoate does not appear to be working as well in BC, and of course we still have these drug issues in the North Atlantic. So unfortunately, Atlantic salmon, as I mentioned, are quite susceptible, and we're trying to look at ways of making them less so, essentially. Now, of the differences in the lice populations, there's two different subspecies between the North Atlantic and North Pacific, and then there's a different species of lice in Chile. Despite all these differences, Atlantic salmon are susceptible to all subspecies and different species of lice, which is quite unfortunate. The positive is that there's variation across different salmonid species. So pink and coho salmon actually display some interesting rejection mechanisms towards the parasite. At early life stages, pink salmon actually have a strong avoidance and predatory behavior towards lice, but they also have a strong inflammatory response. And coho salmon actually has an even more inflammatory immune response to the parasite, which allows them to shed the parasite both in the wild and in Chile, where they grow coho salmon in aquaculture. Infections towards the parasite are quite a bit lower as well. So we're trying to learn from these species as to how they reduce infections to help us in the side for Atlantic salmon. This is an example of a common garden experiment, just comparing some of the different species of salmon. Some collaborative work we've been doing with a group called under a rank called Crispr that's started at Nofima in Norway. And these are size-matched individuals, so very small Atlantic salmon, chum, pink, coho, and between 10 and 35 grams, all exposed to the copepotent first infective life stage of the parasite, and assessing both lice infection level and their impact on these different hosts. And what I'll draw attention to on the graphs on the right side, those are different locations of infection of parasites. So on the top left, there's the infection level on the fins of the fish. And so it's actually the area in which coho salmon, in which Atlantic salmon doesn't have the high infection level. So in this case, you in the first 50 to 72 hours, so the first 3 days of infection, you can see there's a blue higher numbers in the blue line, and that shows coho salmon has the highest infection level on the fins, whereas the other ones are kind of stable. But you'll see over time that those numbers drop drastically in coho salmon. In all the other graphs, the numbers are much higher and the total infection is much higher in Atlantic salmon, and it stays relatively stable. They don't shed the parasite. In comparison, if we look at even earlier life stages of coho salmon, so they generally would go out to sea at about 17 to 18, maybe 20 grams on average size, but there are some smaller coho that will go out to sea. Even at the very small size ranges and only being out at sea for 1 to 2 days, we can see that mechanism for reducing lice, we show on the graph on the bottom here, doesn't really change. And that means that whether they get infected within 24 hours of going out to sea or a month later, the numbers in red and blue there, you see, again, precipitously declining. That, and within 2.5 to 3 weeks, all the parasites are shed. What's interesting is you can actually see this with the naked eye, the infection site. So we've got pictures at the bottom, gross pictures of sea lice attaching to the fin. And if you look at the third picture, it's maybe the easiest to see, where you have a calum life stage. So it's molted on the host, and you see this white or opaque coloration below the parasite. And that's the response of the host. It's epithelial hyperplasia, which eventually overgrows the parasite. And I'll show some other pictures of that, to the point where at 10 days, you can actually see that white or that opaque color on the skin, and the parasite's encapsulated in that skin response. Conversely, it's quite different at the Atlantic salmon. So here we've got histological sections through the skin and fins of infected Atlantic salmon, coho salmon. Atlantic salmon on the left, coho salmon on the right. And you'll see the louse is attached. It's labeled A on the graphs on the left side. And you can see the tissue below. There's really not much of a tissue response in Atlantic salmon. You can see right up to 168 hours post-infection, not a lot's going on. If we look on the right side in coho salmon, again, the louse here is labeled A on the graph B. And you'll start to see that in graph B, there's this skin response beginning to overgrow the parasite. So that's the keratinocyte and epithelial cell hyperplasia. In the bottom pictures, where the red circles are, you can see intensive neutrophil and granulocyte trafficking to the site of infection. So there's an active immune response that occurs quite quickly in coho salmon that we don't see in Atlantic salmon. More specifically, one of our colleagues at Exeter University, Sarah Salisbury, did some RNA sequencing, and this she actually goes through and isolates individual nuclei from individual cells, and then can characterize what are the cell types that are specifically responding to coho salmon at these early times, and that's from 12 hours to 60 hours. And the two major differences that she was able to identify. Now, one of the caveats is that in this technique, neutrophils are not easily isolated, so you don't get a good characterization of neutrophil, which I'll talk about in a different methodology. But here we can see that on the top, Atlantic salmon, there's all these different cell types that are impacted, but mostly T and B cells. So the lymphocyte populations are increased if we compare those to our coho salmon in the bottom graph at the time point. So there's a strong there's a lymphocyte response in Atlantic salmon, but it's not necessarily localized to where the parasite is. It's found through the skin or fin. Whereas in coho salmon, we see this increase in all of these different keratinocytes, these three different populations, and they're all around where the parasite is attached. Two very different responses. Keratinocytes react quickly at all levels. So this is just a kind of a cartoon explaining some of the different expression patterns we see. We have the basal, intermediate, superficial keratinocyte populations, and will enhance extracellular matrix reinforcements that is likely in response to the degradation by the lice, both physical and chemical degradation of the ECM. And then there's a strong motility and proliferation of all populations and induction of immune cell signaling and inflammation. One more thing we've done kind of on a bulk sense, so that's very specific RNA sequencing, but if we do bulk of whole tissues and we compare those on in the infection between coho salmon and Atlantic salmon, we see that there's a number of shared genes that are impacted by infection. However, 80% of those that are shared are upregulated in coho salmon and deregulated in Atlantic salmon. So we're really interested in those because we think that those might be driving some of that response. And many of those are associated with granulocyte recruitment, pathogen recognition, and lectin binding. The final kind of technique that we've used to assess what's going on at this attachment site, and there was some work published also by Sarah and my son Lena Sveen this last year, and it involved doing some cryosectioning of the attachment site to do some spatial transcriptomics. So you can see right where that is attached. We actually preserve the tissue and then go through and do section. And in the pictures A and B, you can see it's at the tip of a salmon fin that the louse attached. And what the spatial transcriptomics allow us to do is measure individual genes at individual cells on the slide. And so we can get an idea of, in the yellow and red kind of quadrants of this slide, are the attachment where the louse is. And in this case, we're able to actually measure neutrophil responses. And we see the strongest pathways impacted are in neutrophil signaling and degranulation. So that kind of gives us our idea of what's going on differently between these two host species, what we think is controlling that strong response to resistance in lice in cohosamin. And this CRISPR-Resist project the idea is to actually see if we can control the resistance by either knocking it out in cohosamin or knocking it in or inducing it in salmon. So I don't know who's all familiar with the CRISPR technique. You may have heard of these dire wolves, or actually gray wolves, that have had CRISPR applied to them to make them look like dire wolves, and they're going down in the US. These two cute little pups down here on the right. It's the same technique. Essentially, it's very simple. It involves the Cas9 nuclease, so an enzyme, and a CRISPR-Cas9 guide RNA. So you have a guide. Essentially, it's almost a prime 25 base pair, 25 base pairs. Is specific to the sequence you want to knock out or cut out. There's a small, what we call PAM sequence, protospacer sequence, that allows the enzyme to attach or land on that primer sequence so it knows where to cut. And then it cuts out chunks of the gene so that it no longer functions. All of ours in Norway were the first to do this in Atlantic salmon back in 2014. And they actually used this technique to knock out pigment genes and create an albino Atlantic salmon. So if you look on the top right, there's the gene SLC45A, and the very top animal, alvin there, albino. And then you can see the two below it. The knockout wasn't perfect, but it created mosaics essentially. And there's examples below of another knockout. And then you see the wild at the bottom. So the WT is the wild type. You can get an idea of how much coloration difference there is with the SLC knockouts. This is great a phenotypic marker, so you can get an idea of, if you're trying to knock out a different sort of function, you don't always know whether you're successful or not. Typically, right? So the idea here is we phenotypic knockout like this albinism or pigment loss, and that'll give us an idea of whether we were successful or not at our CRISPR technique. And we then assume that the gene of interest that we're trying to knock out a similar rate of knockout, but we have to go forward and do sequencing to confirm that. So the fact that knocking in or upregulating gene responses is more difficult, what we've done is started with coho salmon and tried to knock out a few of these genes that we think are very important in the response to lice. So we've chosen 4 targets from our comparative genomics work, as well as we've identified this SL45A pigment gene in coho salmon. It's very similar to that in Atlantic salmon, and use the technique to essentially have an individual immunological or keratinocyte target that we're knocking out alongside our phenotypic knockout, the pigment. So this was not a small feat. We actually had to fly in our gametes from the West Coast of Canada. We had to fly in our research collaborators from Norway, all that little red dot in the middle, which is PEI. And we conducted our experiments in PEI, essentially bringing in the eggs and the milt from coho salmon, fertilizing them, and you have to do the Crispr Cas9 knockouts within a few hours for it to be effective, just as cells are replicating. So this is the group of us in the lab. Our injection team was from Canada, Norway, and Chile. There's my postdoc Manuel on the right, all working injections. And essentially, here is once we've got our Cas9 and our guide RNA ready to go in to manipulate a particular gene, we have eggs, and we actually go on a 25 to 30 degree angle and micro-inject our solution into the fertilizing embryo to knock these genes out. There's quite a bit of variation from the females, eggs on the right. And so we use 3 males and 3 females from 2 different hatcheries in BC. And here's a picture of me just going through and injecting individual eggs. Proof that I actually did some. It wasn't just my postdocs doing all the work. The 4 genes that we looked at used 3 guides for each. So essentially, if we're looking at one particular neutrophil chemoattractant signal, we would knock out 3 locations within that gene to give us a better success of eliminating the function of that gene, and then include our pigment knockout. Expectation is for approximately 20% successful edits on the survivors. Usually we're shooting for about 2,000 embryos. This is from our colleagues in Norway, and you would get about half of those survive. So half that survive, you're looking at maybe 200 successful edits. So the first time we did this, the injection in November, and then January 2024 post-editing. Here you can see the ALVin. You have an albino on the far right. You've got some that there was no successful edits, and then we have some eggs in there as well. And then as they grow up, you actually see them. So here's just an example of some of the pictures of some of the mosaics in the same tank as the edited fish. And then we can actually go through and do PCR of the region, run it out on a gel to confirm. But the best way of determining editing success is using NextSeq, which is a next generation sequencing technique where you get many thousands or even millions of the target sequence to determine how well our knockout was across all the sequences that are available. And as an example for our coho pigment gene, this would be the normal pigment gene on the bottom, and then our CRISPR's successful deletion of 27 base pairs, and we can confirm that when we look through our different phenotypes or different individual ALVin. So just to kind of look at our different targets, we had CSF3R, which is a colony stimulating factor and very important in neutrophil chemotaxis. Mannose receptor 4, which is a chitin binding lectin that we find on neutrophils, and cadherin 26, which is a strong cell factor expressed very highly in keratinocytes. And this is tonacar, who really helped us on much of our editing. And the table just shows kind of the total egg that we received. So 1,200 to 1,500 in each group, and then we have wild type, which were injected. We have our batches from different locations, the total is remaining, and then those that survived to smolt. So we had above 50% with our cadherin survival and our wild type uninjected, but we had slightly lower survival, 36 to 45% in some of the other groups. And our editing success was a little bit lower too. We didn't achieve 40%. We had 9 and 13% successful edits in each of the different groups. So this is what the fish look like following 11 months after they've been edited. And we'll walk through these pictures and kind of the graphs that are associated with them. So what we have at the top are unedited wild type fish. That's what they should look like. Our strong, you can see the strong par marks and dark coloration. On the bottom left is an albino, so there's no pigmentation anymore. And on the right would be a mosaic. I personally kind of like the mosaics. They almost look like a bit of a tiger fish or something. But they don't have as successful a pigment knockout. If we look at our next-seq results, we have an example down in the middle. We have our wild-type controls, and there's 4 different fish shown there. And the colors represent the target. So we have the dark green is our pigment target, and then we have R1, R2, R3, which are the targets for one particular gene. So these are data is for cadherin 26, which was one of our more successful knockouts. On the bottom, you see there really isn't any knockout at all. There's no bar graph except for fish 17. It's a very small, I think it's 2% of the pigment sequences showed a single polymorphism. That's suspecting that that's kind of a natural background. But if we look at our albinos on the far left, you can see there's the 4 bar graphs for 2 different fish. And if the pigment gene, which is dark green on the far right, you can see they're 100% success rate in both those fish. And then both the R2 and R3 targets were almost 100% successful and less successful for our region 1 target. So really success rate for knockout and having edited sequences in those fish. If we look at mosaic, more variable. In the mosaics, you can see that there's anywhere from 20 to 60% success rate in knocking out the pigment. But then our target 2 actually was much more successful. It was 100% in many cases, and then variable between our other targets. So that just indicates to us that although the phenotypic knockout is a good indicator, you're going to have in some cases the gene region that you're trying to knock out may be more or less successful than the phenotypic marker. Another example for CSF3R, in this case, the region 1 that we were targeting weren't successful. We didn't get any knockouts, any edited sequences. And then regions 2 and 3 that we were knocking out were close together. So when we did the sequencing, both showed up together in terms of editing success. We had very good editing success. Orange is the phenotypic marker, and you're seeing more variable phenotypic marker success there. And then we have an example there of in our wild types, we have no sequence showing up that are edited. And then we have normal, quote unquote normal. Those are, in some cases, we have fish that look phenotypically normal, but they were injected. So, and here you can see that there's very little impact on the coloration. There's only maybe 1 or 2% of the pigment was not, but a higher percentage of our other targets. So again, that just lets us know that even though they may be phenotypically normal, they may have a successful knockout in other aspects or the other gene that we need to knock out. So the final kind of confirmation of the impact this would have on sea lice is obviously go through and take these fish through and infect them with sea lice. So we actually did combinations of our gene edited fish along with control fish in the same tank. So unedited and edited fish, so that we could tell they were under the same infection pressure, and then examine both infection level as well as other responses in the fish. So this just gives an example of lice infection the first 7 days. So through the calamus development stages. Blue is the edited, and red and orange is our wild type. So if we look at the edited infection levels, so those are lice per fish on the y-axis, you can see that in the cadherin 26, 40 and up to 50 per fish through those first 7 days. But by day 7 in the wild type, you're starting to see numbers come down. So again, that indicates to us that our editing was successful. We were knocking out something important in fighting off the louse infection. In the CSF3R, again, we also see by 7, even though even in the edited fish, the numbers are coming down, they're down much more to a greater degree in the lice infected, or sorry, in the wild type. Now, the only issue with these graphs are this is before we went through and confirmed whether they were edited or not. So we have a fish that maybe we injected it, but we don't know whether the editing was successful. And where a lot of the variation, especially on the edited fish, lice numbers comes from. When we go through and confirm that they've edited and we compare that to the wild type, we actually see between 68 and 78% reduction in lice numbers in 3 of our 4 gene groups, our Cadherin 26, our CLEC4, and our CSF3R. So that really shows us that the editing did impact lice infection. Something else we thought was quite interesting was the impact on sediment and both on the fins and development on the body. We saw in our edited fish that non-pigmented areas, so those that were edited successfully, had a higher number of lice on them than our non-edited fish. So as we go through days 1, 3, and 7, we see not only are we getting a higher percentage of lice on the non-pigmented areas, but they're occurring more and more on the body. So our infections on the body are increasing from day 1 to 7. That's that blue line. Whereas in the wild type, that number stays quite low and actually goes down from day 3 to day 7. If we look at kind of our original experiment where we're looking at common garden infections, we also see that that in coho salmon, you normally get that very low level in the body, and it goes down from days 3 and 4 and onward, whereas the numbers of lice on the body actually increase on Atlantic salmon. So it's kind of similar to what we're seeing here in the edited coho salmon. I'll just wrap up with a couple other ways of showing that we observe success in our knockout. This is, we actually went through into quantitative PCR of the targeted gene in our wild type and our in our edited fish. So on the far left of these graphs, we have gene expression relative to a reference gene at days 3 and 7 post-infection wild type. And you can see from days 3 to day 7, you have a strong induction of this lectin gene in our wild type. If we look to the next two, the 2 in the orange, we just see a flat line essentially. So, and if we look on in the table, we see that the kind of expression level at 3 and 7 days in our control goes from about negative 0.5 to positive expression at almost 1.5. But our target, CLEC4, is negative at 3 days and actually stays negative. So again, it's having a different expression profile than what we see in our control fish. And we compare that to other studies on coho salmon that are infected. And you can see here by Braden et al., early on, again, days 1 to 7, the expression of many of these lectins turns on. So that's why you see all this pink and reddish coloration. That's induced expression in the early days of infection. Similarly, all the other genes, Cadherin 26, we actually see from day 3 to day 7 in the wild type, it being the expression decreasing, but not to the same extent in the knockouts. We have much greater reductions in the knockouts. So again, the profiles are changing for the genes that we targeted. And the last thing I'll mention here, not only is the expression of the targeted gene impacted, but also seeing changes in cell trafficking in those infected fish. So this is work that Javon Lowe, our grad student, recently did, just looking at different cell populations in these coho salmon. And if we look at the wild type on the far right and we compare that, to, for example, to CSF on the left, we can see very different profiles. So we commonly get about 30 to 40% of the peripheral blood leukocytes in these coho salmon are neutrophils, and about 50 to 60% are lymphocytes. In our CSF impacted fish, we actually see it almost close to 100% by day 7 are lymphocytes. So very different impact, or very strong impact on neutrophil populations, which is what we expect since these are neutrophil markers. We're actually getting neutrophil, they're important in neutrophil trafficking. And so we're actually getting different cell types responding because the neutrophils apparently aren't working as well. In cadherin, on the far right there, we also don't see as much of a difference in the lymphocytes and the neutrophils. They're a little bit lower and a lot of variation, of course, but we are seeing a change in the monocyte population. We actually have almost 2 to 3 times the levels of monocytes in circulating in these fish. So we have more work to do here, but we really are seeing differences in the genes that we're knocking out and the downstream cellular trafficking and the lice levels. So in conclusion for this kind of these techniques and understanding of controls of sea lice infection, we've seen that knockout success in that first generation is quite variable. The guide RNA target success can be quite variable, so some targets work better than others, and even can be better than our phenotypic marker. But those with successful edits did significantly increase calamus lice stages, so there is a relationship between these targets. Some are better than others, like cadherin 26 and CSF3R. We had really strong or really high levels of calamus. And that there's an impact on gene expression and cell trafficking, and that the mosaics may actually also shift settlement and development patterns based on only having expression in certain areas and not in others. So analysis of this work is ongoing. We have large-scale RNA sequencing that we're doing of the edited fish, both at the systemic and the localized levels. We're looking at what are the most important types of edits. So those that confer a frame shift are likely going to have a greater impact, but we haven't confirmed that. We're still looking through some of our sequencing data. And now we've actually begun a second experiment where we're going to breed our F0s and generate an F1 knockout to get a higher success rate of our knockouts and see whether there is improved success in the F1 generation. Of course, the goal of this was in the end to get to a point where we see genes that are important in impacting lice, and can we do that in Atlantic salmon? So we actually have now a few of these targets got CRISPR knock-ins or induced expression Atlantic salmon. That's begun, but Atlantic salmon takes longer to grow. So it'll be a little bit longer before we can actually do the testing with sea lice. The last thing I'll say, the goal of this research is really to identify important controls of lice infection. Now that can be used in breeding programs to look at particular targets and try and find salmon broodstock potentially have higher expression of these genes. We're not using it as a method that we think that the fish would be edited and released in the wild. I think we're not condoning that. There's definite ethical issues with doing something like that. But as part of our research, we actually have collaborators in Norway and Australia that have looked at modeling gene-edited flow if they were released in a place like Norway. So there is some analysis going on and just showing that if edited fish were used in the north of Norway, the passage of gene flow is actually not as strong as if they were released in the south. But that again isn't a goal of our work, but we think it's still a good idea to kind of look at if different potentials for the ethical use of these kinds of techniques. This is our collaborative group. I have many of them to thank, but I will stop now and hopefully we have some time for a few questions. Thank you for your attention.
That was great. Thanks, Mark. Really, really interesting research. So we'll open the floor to questions now. So if anyone has any questions, just raise a little hand on your screen and you should be able to unmute and ask your question. Give it a second, see if anyone asks anything.
Hopefully I didn't go through it too fast and it made sense.
Yeah, we haven't had very many question askers the last couple of, oh, there's one coming in, great. Okay, go ahead, Peter. You can ask your question now. Peter Salonis. Peter, do you have a question? Peter Salonius? Just make sure you allow your computer to access your mic there and you should be able to unmute.
There, can you hear me now?
Yep, we can hear you.
Hi, Mark. I've been concerned about spillover into the wild population for a long time. I'm sure you have too.
Yes.
And what I've come up with as an interim solution is the fact that the bay management system has fallowing of some of the farms every year, but you've always got a population of sea lice in the water emanating from the farms. So what I've come up with is suggesting that the all of the farms in New Brunswick are fallowed on a particular fall. So they harvest everything, they sanitize the nets, and they leave them fallowed until the next spring, and then they restock them. So we have at intervals seawater which is essentially free of aquaculture or origin threats.
Yeah.
And that may be a little hard to sell to the industry, but as a colleague of mine said, well, you might have some unemployment in the field, and I said, well, tourism, road construction, and a lot of other industries have layoff periods that are supported by unemployment insurance, and maybe it's time we gave wild salmon a chance, because the smolt are very vulnerable. And there's always been a population of sea lice to greet them as they hit salt water.
Yeah, no, you're not totally wrong. And I think the industry has moved towards all in, all out in certain regions and certain bay areas, and then that bay area isn't restocked. However, there's always still some potential in the whole. If you're looking at New Brunswick, there's the Bay of Fundy is a large body of water with a lot of flushing, right? So there is the ability for the lice to move around, even if they're maybe not maybe if they're in different bay management areas, to still circulate. And I think the understanding of how even under fallowed conditions, we still don't have as good an understanding of where these lice are during these fallowing events as you think we would at this point, right? Like in cases where there's not a lot of wild salmon in the area, and we're still having large numbers of lice available to the fish when they do get restocked. So I agree that we do have to come up with a better system of preventing and reducing that infection pressure. And I think it's like anything, there's not going to be one solution, right? Medicines were never a single solution that were going to work forever. The new mechanical treatments work can be quite successful, but they also are a welfare concern for the fish. And then there's the new cage designs, which can be quite good as well. And I think those are coming more and more online. I don't know of the snorkel cages or things like that, whether they've To my knowledge, I don't think they've been used in Atlantic Canada yet, but I think it's a combination of, addressing both kind of cage design, intervention strategy, genetics, and also moving the fish around and stocking at different times, and shortening that window too. I think there's definitely a movement to stocking larger smolt and having them in the sea cages for shorter periods of time, so that the infection level doesn't get so bad. But we're definitely not there yet. Got a lot of work to do.
One thing I was impressed with, Mark, was work in Norway on the viability of sea lice in cold salt water, and the distance that the as yet non-infective lice could travel. And I think it was something like 100 kilometers. So, and then I started to think about the Bay of Fundy and the flushing that goes on, and it occurred to me that the bay management system didn't accommodate that sort of flushing.
That kind of distance.
And the only way I could see around it was to just shut them right down and then restock them. If it didn't lower the lice pressure entirely, it certainly would decrease it. With
possibly,
with
and
synchronous fallowing.
Yep. Yeah, and I think the synchronous fallowing does occur. I think it's I just don't know to what level of the entire industry would being removed. And that's just not an option, right? Like if you look at in British Columbia, they've shut down areas in the Discovery Islands for 2 years, and the lice numbers are just as high now in those areas as they were during farming. The dynamics are completely different there, but it just speaks to the fact that we really need to make sure we have a good handle on how these parasites are circulating before we go too far in some of our management strategies in terms of trying to lower what we think is lowering infectious pressure.
I guess my feeling was that the industry already is submitted to serial fallowing, so they're used to that, and to fallow everything all at once in a fall and restock in the spring shouldn't be a big step. Now there is one thing about supplying the market, but organizations like Cook have access to stuff for the commercial trade all over the world. They don't rely on the Bay of Fundy entirely.
That's true.
Anyway, this was really interesting, and I hope you get someplace with the CRISPR situation. But for now, I'm going to stick with the business about trying to alter the fallowing practice.
Yeah.
Thanks very much.
Thank you.
All right, that's great. Thanks, Peter and Mark. It doesn't look like we have any more questions. If anyone does have a question later on for Mark, feel free to reach out to the foundation, and we can pass along your question to him. So I have recorded this session. It will be available on our YouTube page later today. I would like to remind everyone that our next webinar is going to be next Wednesday, February 11th, with Natalie Koopman as part of our Student Spotlight series in partnership with the ASRJV. And the title of that webinar will be Assessing the Causes of Atlantic Salmon Declines in Cape Breton and the Efficacy of Smolt to Adult Supplementation for the Restoration of Endangered Populations. So again, a big thank you to Mark and everyone who participated today. Have a great rest of your day, everyone.
Thank you for having me. Thanks, Kristen.
Bye-bye.